Introduce you, we'll get started. Okay, why don't we get started? Thanks for coming out, everybody. A morning seminar, unusual time, but we're working on balancing a lot of schedules here. So it's a pleasure to re-welcome Dr. Andre Ballesteros-Tato from UAB. Andre was here about a year and a half or so ago and gave a seminar. Some of you may have heard, so he's back, which is great. We're delighted to have him here. Andre did his PhD in molecular biology at the Autonomous University in Madrid, Spain, and in the National Center for Biotechnology. He then made a move to the United States, initially to a very beautiful place, Lake Saranac, New York, to be at the Trudeau Center for a short period, a center that I knew actually very little about. I had heard of it, but apparently it was quite a hotbed for immunology research and exciting place to be. He handled the winters up there and decided he needed to be colder, so went up to Rochester, New York, to the University of Rochester with his postdoctoral advisor and spent several years working there doing his postdoctoral training. And then moved to UAB, University of Alabama at Birmingham, where he joined initially as a postdoc and then a research assistant professor and then an assistant professor in the Division of Clinical Immunology and Rheumatology in the Department of Medicine at UAB Medical School. And for those of you who don't know the field that well, that particular place, UAB, Division of Immunology and Rheumatology in Medicine, is a really storied and strong, one of the more outstanding programs. I know that historically because I used to be there years ago. So he's embedded in a really strong group there where he's now an assistant professor. Andre serves on a number of editorial boards, including on Scientific Reports, and serves as an advisory editor for arthritis and rheumatology. And he also is a recipient of the Jim Pittman Endowed Scholar Award at UAB. Jim Pittman was the dean of the UAB Medical School for a number of years. It is usually credited with having built the UAB Medical Center into one of the premier medical centers in the world. And so I know that's a prestigious position to get, or an award to get at UAB. Andre has published on T follicular regulatory cell response to a variety of signals. He's particularly looked at cytokines like interleukin-2 and their role in a number of issues, including in autoimmune disorders and potential translational and clinical implications of low-dose IL-2 treatment in autoimmunity disorders. He has used an influenza model to specifically look at T follicular helper cell responses and their regulation by pathways, including the FOXP3 pathway and regulatory T cells, and gone very deeply into understanding effects of priming on T follicular helper cells by dendritic cells as well. So he's added a lot to the literature. I'm not an immunologist, obviously, but I think this stuff looks like really solid stuff, and his papers have been well regarded and recognized by others in the field, and some of these findings have been used by others to advance the field further. In addition, he has active collaborations with our own Ken Astrike and some of the students in the lab here as well. And also, I should mention that his wife, who's here visiting as well, Dr. Leon Ruiz, who I hope some of you will come and hear as well tomorrow, has been a collaborator in a number of projects, although has her own independent research program as an immunologist as well that we'll be hearing about. So without further ado, I'd like to welcome Andre for his talk. Thanks for being here.
Can you hear me? All right. Thanks, Mike, for the introduction. I'm glad to be here, and I'm very excited about my talk today because this is a project that started in my lab like 2 years ago. Graduate student started this project, and it was one of those that is a high risk, high reward project. When Megan started my lab, I told her, I have this idea. It's a crazy idea. You might be doing your thesis for like 10 years because it might not work. But she is a force of nature, and in 2 years you will see how much she was able to advance. And this is the first time that I present this data outside UAB, so I hope you like, and I would really love to hear back your feedback. So today I'm going to talk about CD8 T cells. And so CD8 T cells is a subset of T cells, and they have the unique ability to identify, and then once the cells are identified, to kill cells that are infected by intracellular pathogens. That means, for example, cells that are infected by virus or infected by bacteria. And they also pretty good are recognizing and killing tumor cells. And indeed, we know that CD8 T cells are fundamental for protective immunity because in the absence of CD8 T cells, protective immunity to pathogens or protective immunity to tumors is strongly compromised. And indeed, for example, you probably know about the latest immunotherapies for tumors that are based on checkpoint inhibitors like PD-1 or CTLA-4. Well, the mechanism underlying those therapies is actually waking up the CD8 T cells so they can go look for the tumor and then kill them. So it's really important that we understand the mechanisms that controls the generation of a good CD8 T cell response, so we can manipulate it, the CD8 T cell response in a way that we want, so we can generate better immunotherapies against tumors and better vaccine strategies to intracellular pathogens. But before the CD8 T cells go and look for the targets and kill them, they need to be pre-activated, and that's what we call priming. And priming happens usually in the secondary lymphoid organs. The secondary lymphoid organs are, for example, the spleen, which drains the bloodstream, or the lymph nodes, which are connected to the tissues. And there's where the CD8 T cells get first activated. And in order to be activated, they need to interact with dendritic cells, which is what you have here. So dendritic cells is a subset of innate cells that have the unique ability to be activated by pathogens. And so the way it works is that dendritic cells, they express a broad range of pathogen recognition receptors. So when they close encounter a pathogen, dendritic cells get activated. And as a consequence, they start to produce pro-inflammatory cytokines. They start to upregulate costimulatory ligands, and they start to capture antigens from the pathogens and present these antigens complex to class 1 molecules. So when these activated dendritic cells encounter the CD8 T cells, what's happened is that dendritic cells provide the CD8 T cells with inflammatory cytokines, provides them with an antigen that activates the TCR. This is the T cell antigen receptor that the CD8 T cells have, and they provide them with costimulatory ligands, CD28 and CD27. And those three signals are known as signal 1, signal 2, and signal 3. And those three signals are the signals that CD8 T cells need to be activated. Basically, it's like they need these three signals to awake and say, okay, there is a pathogen out there, I have to go and kill it. And so what you have here is a representation of how a tissue is connected to a lymph node. Basically, you have your lymph node, and this is the tissue. Let's say, for example, I'm going to talk about influenza. This will be the airways. The airways are connected to the lymph nodes by the afferent lymphatics. So cells can go from the tissue into the lymph nodes through the afferent lymphatics. And this is a one way. So cells can go from here to here, but they cannot get back into the tissues. To get back into the tissues, they have to leave the lymph nodes through the efferent lymphatic vessels that are connected to the blood vasculature, and their cells can go back into the tissues from there. So basically, what's happened is that CD8 T cells are activated here, they get into the bloodstream, and from the bloodstream, they can get into the tissues where they find the infected cells and kill them. And so I told you that to be primed or to be activated, CD8 T cells need to interact with dendritic cells, and this usually happens in the lymph nodes. And there are two subsets of dendritic cells in the lymph nodes, migratory and resident dendritic cells. So resident dendritic cells are dendritic cells that differentiate locally here in the lymph node from precursors that enter the lymph node from the blood. So basically, those dendritic cells are in the lymph node, never been in the tissue before, and will never be in the tissue. And those are important, for example, in the case of systemic infections, where the pathogen can get into the bloodstream, and so the pathogen can get access to the lymph node. In that case, the resident dendritic cells are going to see the antigen, are going to present antigens to the CD8 T cells, and going to be priming CD8 T cells. And then a second main subset of dendritic cells are migratory dendritic cells. And those are dendritic cells that are in the tissue, they differentiate locally in the tissue, and if they see an antigen, they're going to be activated. And if they're going to be, after activation, they're going to have the ability to enter the afferent lymphatic vessels. And from there, they migrate into the lymph node, and when they encounter CD8 T cells, they're going to prime the CD8 T cells. So a few years ago, we were interested on which dendritic cells are responsible for priming CD8 T cell responses to influenza, the resident dendritic cells or the migratory dendritic cells. And so we start looking at that, and I'm not going to show you any results. I'm going to show you the summary of the conclusions of this work. What we learned is that we used an influenza infection model. in mice. And the way it works is that we intranasally infect the mice, so the virus go into the airways and the lungs and induce a productive infection. And what we learned is that when we do that, if we infect mice intranasally, the virus do not get from the tissue into the lymphatics. So if you look into the lymph node, you don't find virus, and you don't find free antigen from the virus. What that means is that if we put virus or antigens here, those antigens do not get into the lymphatics. And that makes sense because you don't want to have a systemic spread of the virus. So the lymphatics are not very permeable for the pathogen. And as a consequence, because the antigens or the virus do not get into the lymph node, the resident dendritic cells in the lymph node do not have access to the antigen, and therefore they are not activated and they don't present antigen. So if you take resident dendritic cells, they don't prime CD8 T cells in this model. Instead, what's happened is that when you infect the mice with influenza, the dendritic cells that are in the tissue, they see the virus, they get activated, they enter the lymphatics, they migrate into the lymph node, and once in there, they encounter naive CD8 T cells and they activate them. And indeed, if we eliminate dendritic cells from the tissue, or we have dendritic cells that cannot migrate from the tissue into the lymphatics, the CD8 T cells are not prime. Other people have looked at similar models in which the pathogen do not spread systemically, meaning that the pathogen do not get straight into the blood. Similar model applies. In those models, usually the dendritic cells that are in the tissue are the responsible for priming the CD8 T cells responses in the draining, whatever the draining lymph node is. For example, in case of influenza, it will be the lung draining lymph node. If you are thinking in the skin, there will be another lymph node, so on and so forth.
Is this just for influenza or for any type of virus?
So this model applies for pretty much any immunization model or infection model in which the antigen or the pathogen do not get into the bloodstream. Okay. So pretty much all the mucosal infections. So these have led to a paradigm. And I'm going to use this cartoon a lot through the talk, so let me guide you through. So what you have here is a representation of the tissue. So I'm going to be talking about influenza, so this tissue will be, for example, the airways or the lungs. The tissue is connected to the draining lymph node. In the case of influenza, the draining lymph node is the mediastinal lymph node, it's right under the heart, and it's the tissue, and the lymph node is connected to the lymphatic vessels. So cells can go from here to here using this connection. And cells only go in this direction. Cells do not go back into the tissue through the lymphatics. Cells that are in the draining lymph node can get out of the lymph node through the efferent lymphatic vessels. The efferent lymphatic vessels are connected to the blood circulation through the thoracic duct. So cells can get from the tissue into the draining lymph node, and activated T cells can get out into the blood circulation. And from the blood, they can go everywhere. They can go back to the tissue, they can go to the spleen, or they can go back into other secondary lymphoid organs through the high endothelial venules. So the current paradigm says that if you have an infection in a tissue, let's say influenza in the airways, migratory dendritic cells that are in the tissue are going to be activated. They're going to capture antigens. They're going to enter the lymphatic vessels. They're going to migrate into the draining lymph node. And because they are activated, they produce pro-inflammatory cytokines. They have all these costimulatory molecules. They have the antigen. And as a consequence, if they encounter CD8 T cells that are specific for that particular antigen, the CD8 T cells are going to be activated. And this usually happens very quickly after infection. Usually within these 6 to 24 hours, activated dendritic cells migrate from the tissue into the draining lymph node. Then during 3 days, the T cells proliferate. And they proliferate, they get activated, they start to acquire effector function. That means the capacity to kill. These dendritic cells, usually they have a very short lifespan, so after 3 days, they are gone. But the CD8 T cells, after 3 days, they are ready to go to the tissue and start killing infected cells. And what's happened is that they enter the efferent lymphatic vessels. From there, they get into the circulation. From the blood, they go back into the tissue, and there they start killing infected cells. And also, additionally, you have some cells that are activated, and instead of going to the tissue, they're going to go to other secondary lymphoid organs, like the spleen or the lymph nodes that are not directly connected to the lymph node. And that's going to provide systemic immunity.
Are those cells being targeted already with the signal, or is that just a random distribution of cells?
No, those cells are all these cells are going to be antigen specific, so they're going to be specific for the pathogen.
But I mean, one that goes to the spleen versus to the tissue.
So there are different signals that are, we call homing signals,
Right.
that is going to direct them into different locations.
What determines the one wayness of the afferent vessels?
Excuse me, say it again.
What determines the one wayness of the afferent vessels? Are they valves or?
Yes, so there are bulbs that the lymph flow goes this way, and because there are bulbs that basically pump the lymph up.
So it's purely mechanical.
It's pure mechanic. And then there are active mechanisms for which the cells can get into the lymphatic vessels, and we will talk a little bit about that. So it's not like an open door where everything can get in. You have to actually transmigrate the endothelium. So if you think about this model suggests that you have a very local immune response, so dendritic cells that get activated in a tissue, migrate to the regional lymph node that is directly connected to the tissue, and then the CD8 T cells get activated. Some of them go back to the tissue, some of them go back and spread systemically. So if you think about, you go from a very local immune response to a very systemic response, but everything starts in the draining lymph node. And that's a great paradigm and explains a lot of the events that we see after infection, but there are some things that cannot be explained by this paradigm. For example, if we look at a whole immune response after influenza or even any other infection, and we look in the spleen, we have germinal center B cells. And in the germinal centers, B cells undergo somatic hypermutation and affinity maturation, and that's what gives you antibody responses, right? The high affinity antibody responses. But that requires the constant presence of antigen. If we don't have antigen, the germinal center B cells cannot develop. So if we have germinal centers in the spleen after influenza infection, that means that we have antigen in the spleen. And also there are some papers that suggest that when you look closely, some CD8 T cells might be primed in the spleen. And this doesn't fit with the current paradigm, because if you remember, I told you that first, the tissue and the spleen is not connected by the lymphatic vessels. So there is nothing that connects the spleen and the tissue. And also, I told you that the virus or the antigens from the virus do not get freely into the lymphatics or do not get freely into the circulation. And also, I told you that dendritic cells die after 3 days of being in the lymph node. So if we have antigen in the spleen, we don't know how the antigen gets there. And if we have antigen in the spleen and CD8 T cells are primed there, we don't know what are they doing or what are the contribution of these cells to the overall immune response. And this is the kind of questions that we want to answer today. So if we have the antigen, how is the trafficking of the antigen, how the antigen gets there? If we have priming, which dendritic cells are priming CD8 T cell responses in the spleen? And if we have CD8 T cell responses in the spleen, what are they going to do? What is their relative contribution to the overall response? So the first experiment that we did is a very simple experiment. We thought, well, if we have antigen, the antigen is going to be presented to the CD8 T cells by dendritic cells. So let's going to look at which dendritic cells we have in the spleen and then compare it to the dendritic cells that we have in the lymph node. So what we did is we infect mice with influenza. Again, this is an intranasal infection, so it mimics what happens in humans. You have an intranasal colonization of the virus. It's going to infect some epithelial cells. The virus is going to divide. It's going to proliferate. After 3 days or so, you're going to have CD8 T cells going into the lungs. It's going to start clearing the virus, and after 7 to 10 days, the mice are fine again. So we infect the mice with influenza, and then 3 days later, we look at dendritic cells in the lymph node, in the draining lymph node, and in the spleen. So when we look in the lymph node, we use a very easy gating strategy. Basically, we gate out T cells and B cells, and then we use class II and CD11C to identify migratory and resident dendritic cells. So if you look at here, we have 2 populations of dendritic cells. This population right here, which is CD11C high and express medium levels of class II, are the resident dendritic cells. And this population here are the migratory dendritic cells. Those cells express high levels of class II and express intermediate levels of CD11C. And another marker of migratory dendritic cells is CCL7. Usually, migratory dendritic cells express high levels of CCL7. And this is because this is the chemical receptor that dendritic cells need to enter the lymphatics. So we use the same gating strategy, and then we look in the spleen, and we ask which dendritic cells we have. And this is the spleen. And as you can see, it's quite different from the lymph node. In the spleen, the majority of the dendritic cells look like resident dendritic cells. They are CD11C high, they express medium levels of class 2, and they are CCR7 low. If I go back to the lymph node, these are the resident dendritic cells in the lymph node. Here you have the spleen. So the majority of them look like resident dendritic cells. But we have a few of them that look like migratory dendritic cells. They express high levels of class 2, they express intermediate levels of CD11C, and they are CCR7 positive. So they look like the dendritic cells that we have in the lymph node, and we know they are coming from the lungs. So the next question we ask is, well, is there any possibility that some of these dendritic cells are actually migratory dendritic cells that are getting into the spleen somehow from the lungs? And to answer this question, we did a labeling and tracking experiment. So let me show you how this works. So in these experiments, what we do is we infect mice with influenza intranasally, and then we administer CFSC intratracheally. So CFSC is a labeling agent. And what's happened is that when we do that, when we add the CFSC intratracheally, all the cells in the airways are going to be green, all the cells. If we wait long enough, that means 16 hours or so, we know that some of these dendritic cells are going to migrate into the draining lymph node through the lymphatics. So we can use flow cytometry to ask which dendritic cells in the lymph node are green. And those that are green, we know that are coming from the lungs. And here you have an example of this experiment. This is a lymph node. We use our gating strategy, CD11C, class 2. And if you look at here, if we gate on these cells, those are the migratory dendritic cells. In mice that receive PBS, we don't have any green cell. If we administer CFSC, we have like 30% of the cells are CFSC positive, meaning that they are coming from the lungs. If you look at the migratory dendritic cells, which are here, you don't. That's one of the ways that we can actually identify migratory dendritic cells. So we did this, but we look in the spleen. So let me guide you through the experimental design. We infect mice with influenza. 48 hours, we have 3 groups. One group received PBS intratracheally. This is our negative control. This is what is going to show us the background. Then we have our positive group, which received CFSC intratracheally. Okay, so all the cells in the lungs are going to be green. And then we have a group C, which is kind of a positive control, in which the CFSC was given retrorbitally, so intravenously. So in that case, all the dying agent is going to get straight into the spleen, so everything should be positive. So here you have the negative control and the positive control. So if we gate here on the migratory and the resident dendritic cells in the spleen, we don't have any positive cells in the PBS treated group. Pretty much we have a high frequency of positive cells in the mice that receive CFSC retrorbitally, no matter what they were resident or migratory. And then we look at the CFSC treated mice. And as you can see here, only the migratory dendritic cells were positive for CFSC. What this result shows is that this tiny population of cells that were class 2 high, CD11C intermediate, and express high levels of CCR7 were actually coming from the lungs after infection. So this is how we think this is working. We have dendritic cells in the tissue, and then somehow we end up with some of these cells relocating into the spleen. So of course, the next question we want to ask is, how do they get there? Right?
Can I ask you a quick question? What is the time frame for all of those, for the infection, for the migratory C cells to get the CD cells to get to the spleen?
Yeah. So when you infect mice, it takes about 6 hours for the dendritic cells to get activated, and they start migrating into the lymph node right away. So the way that we do the experiments is when we want to assess the migration, we give them CFSC on a particular time point, and then we wait 16 hours. And in that time frame, we know that the dendritic cells already go from the lymph node, from the airways into the draining lymph node. And then we have migration for the first 5 days. So for the first 5 days, you have continuous influx of dendritic cells from the lung into the lymph node, and then decays.
So is there any other, when you infect with the virus, could any other organs be infected other than the airway?
No, when you infect with influenza, the only cells that are getting infected are as club cells that are in the airways. So there is no, for example, if you take influenza virus and you inject it in the skin, cells are going to be infected, but the virus is not going to divide, and you're don't going to have a productive infection.
And you're injecting this kind of marker intratracheally, you said?
So the virus goes intranasally, so they basically breathe the virus,
Yeah.
and this label, the CFSE goes intratracheally. So we put it straight into the airways. So only the cells that are in the airways are going to be in contact with the dye. So all those cells are going to be labeled.
So that's clear at this point, but did you just kind of have a secondary confirmation
Yeah.
gate for that like marker?
Yes, yes. I'm going to show you more confirmatory experiments to show pretty much the same thing using a lot of experimental approaches. That's a pretty good question in there. So we know that somehow they get from here to here, right? So how do they get, if they move to the spleen, how do they get there? We know that there is no connection, lymphatic connection between the tissue and the spleen, so that's not an option. An obvious option will be that somehow the dendritic cells get from the tissue into the blood, and from the blood they will get straight into the spleen. We know that although this is a theoretical possibility, it's very unlikely. There are not mechanisms by which immune cells can get into the circulation from the tissues. Okay. But it could be. And the other option is that dendritic cells get into the lymphatics, migrate into the draining lymph node, and then somehow, if they don't encounter T cells, get out back into circulation, and then from there to the spleen. So what we did to test these two possibilities is we decided to block the capacity of dendritic cells to enter the lymphatics. So if they need to enter the lymphatics and migrate into the lymph node to get to the spleen, if we block this, we will lose dendritic cells in the spleen. Go ahead.
So by no mechanisms to get into the circulation, there's just no way that the cell can actually get across, like the...
Correct. Unless you have massive damage.
And is that affected by whether or not it's carrying an antigen, or is it the same for just this dendritic cells alone?
So looking at other immune cells, T cells, B cells, dendritic cells, when you look at how they can migrate in the tissues or from the tissues, they can be in the blood and get into the tissues. There are some mechanisms that allow that, but there are no mechanisms that allows them to be in the tissue, get back into the blood, because they need to cross the endothelium. But we don't know the mechanism, but it could be possible that there are mechanisms, right? Okay, so going back, we decide to block the capacity of dendritic cells to get into the lymphatics. So in order to get into the lymphatics, the way that dendritic cells get into the lymphatics is that they are in the tissue. This right here, this will be a tissue, the airways, for example, and the lymphatics get into the tissue, and the lymphatics connect the tissue with the draining lymph node. So to get in there, dendritic cells upregulate CCR7. This is a chemokine receptor. The ligand for CCR7, which is CCL21, is highly expressed in the lymphatic vessels. And so if you have a cell that expresses high levels of CCR7, they're going to be attracted into the lymphatics. They're going to enter the lymphatics, and then with the flow, they're going to go into the draining lymph node. So what we did is we did bone marrow chimeras in which half of the dendritic cells will be white type and half of the dendritic cells will be CCR7 knockouts. They will not be able to enter the lymphatics. So I don't know if you're familiar or not with bone marrow chimeras, but this is what we did. We took white type recipient mice, and then we irradiate them. And what we do when we irradiate mice is that we deplete all the bone marrow precursors. So if you don't do anything else, after 3-4 weeks, the mice start to die because they don't have competent precursors from the bone marrow, so they cannot reconstitute their immune system. So what we did is we irradiate the mice, and then we reconstitute them with a mix of a 50-50 mix of bone marrow from white type or CCR7 knockouts. And we can tell from which bone marrow every cell comes, because the white type bone marrow express this marker, CD45.1, whereas all the cells derived from the CCR7 knockout bone marrow are going to be CD45.2. Let me show you how this works. We wait 2 months, then we infect them with influenza. We give them CFSE intratracheally, and then 72 hours, we look at the different dendritic cells population in the spleen, lymph node, and lungs. And I think it's going to be easier to understand once I show you the results. So if we look at the lungs, here you have your dendritic cell population, and as you can see here, 50% of the cells are CD45.2. Those cells are the CCR7 knockout. The CD45.2 negatives are the wild types. We did a bone marrow that is 50-50 wild type CCR7 knockouts. So if we look in the lungs, we have a 50-50 ratio CCR7 knockouts wild type. That makes sense because CCR7 is not required to get dendritic cells in the lungs. If you look in the lymph node, you get on migratory dendritic cells, so meaning class 2 high CD11C intermediate, you get on your CFC positive cells, and here you have that almost 100% of the dendritic cells are wild type. You don't have any CCR7 knockouts. That makes sense because we know, it's been published 1,000 times, that in order to get from the lungs into the lymph node, they need CCR7. Then we look at the spleen. If we get on the resident dendritic cells, again, we have a 50-50 ratio. That makes sense. We know that CCR7 is not required to get into the spleen. If you get on the migratory dendritic cells and get on the cells that we think are coming from the lungs, you only have wild type cells and not CCR7 cells. So expression of CCR7 in dendritic cells is required to get dendritic cells into the spleen. So this model suggests that when we have infection, some dendritic cells get activated. They don't get into the blood circulation. What's happening is that they upregulate CCR7, they enter the lymphatics, they go into the draining lymph node, and from there they get out. And once they are out in the blood circulation, they can get back to the spleen because everything goes into the spleen. So to prove this model, we decide to block the capacity of dendritic cells to get out of the spleen, of the lymph node, and ask whether or not that prevents the homing into the spleen. So the mechanisms that control the egress of immune cells from the lymph node into the blood has been studied for T and B cells. And what we know is that when T and B cells are activated, they upregulate the S1P receptor. And the ligand for S1P receptor is S1P, and this is highly expressed in the efferent lymphatic vessels. So what's happening is that you have an activated cell that expresses high levels of S1P receptor. They're going to be attracted into the efferent lymphatic vessels. Once they enter the lymphatic vessels, they're going to enter the thoracic duct and from there to the circulation. That's the way they get out. There is a drug that is called FTY720. That what this drug does is that induces the internalization of S1P receptor. So once the T or the B cells internalize the S1P receptor, they can no longer leave the lymph node. So if you treat mice with FTY720, and even humans because it's FDA approved, cells get stuck in the lymph node and they cannot get out. And we know dendritic cells express all the S1P receptors, S1P receptor 1, 2, 3, 4, and 5. So it makes sense that similar to T and B cells, they might need the expression of S1P receptor to get out of the lymph node. So what we did is we infected mice with influenza. Then we did our migratory labeling and tracking experiment. Basically, 48 hours after infection, we give them CFSE, or we give them CFSE plus FTY. And then 24 hours later, we look for migratory dendritic cells in the lymph node and the spleen. So if you look in the lymph node, we found similar frequencies of migratory dendritic cells in the control or in the treated group. That makes sense because you don't need S1P receptor to get into the lymph nodes. So we shouldn't be affecting the presence of migratory dendritic cells in the lymph nodes. But when you look into the spleen, as you can see here, this is our control group, this is our treated group, we have fewer migratory dendritic cells in the spleen. And you have here the frequency, and you have here the numbers. If you look at resident dendritic cells, we have the same numbers. So this indicates that dendritic cells go from the tissue into the draining lymph node, and from the draining lymph node into the blood. And if we block the capacity of dendritic cells to get out of the lymph node, we lose our migratory dendritic cell in the spleen. So this shows that dendritic cells migrate. But the next question is, in addition to migrate, do they bring the antigen with them? Because if they migrate and they don't have antigen, then it's irrelevant, right? So we decide to look at whether, in addition to migrate, they also bring the antigen with them. So this is quite a complicated experiment, so let's see if I can guide you through. We infect mice with influenza. 48 hours, we give them CFSE intratrachely, so all the mice receive CFSE intratrachely. So all the cells in the lungs are going to be green. And then we have two groups. In one group, we give them OVA intratrachely. So they have CFSE and OVA. OVA is ovalbumin, is a protein. And this OVA is conjugated with Alexa 647. So using flow cytometry, we can tell which cells are CFSE positive and which cells are also OVA positive. And then in the group B, we give them OVA 647 in the foot pad. So the foot pad is not connected to the lung draining lymph node. I'm going to show you the results, and I think it's going to be easy to understand what we are doing here. So what you have here is the mediastinal lymph node. This is the lung draining lymph node. This is the popliteal lymph node. This is the lymph node that drains the foot pad. And then we have the mesenteric lymph node. This is in the gut. And it's not connected to the lungs, and it's not connected to the foot pad. So if you look at here, this is the mediastinal lymph node. This is the lymph node that drains the lungs. If we give them CFSE, we have CFSE positive cells in both groups. That makes sense because the CFSE was given intratrachely, and this lymph node is connected to the lung. So those are the lung migratory dendritic cells. If you look at the group that received OVA intratrachely, we have OVA positive cells. So we have cells that were in the lungs, acquired the OVA, and migrate into the lymph node. If the OVA was given in the foot pad, we don't have, because the OVA was given in the foot pad, and the lymph node, this lymph node is not connected to the foot pad. When you look in the popliteal lymph node, we don't have CFSE positive cells, because the popliteal lymph node is not connected to the lungs. We don't have OVA in the mice that receive OVA intratrachely, because in this case, the popliteal lymph node is not connected to the foot pad. But we do have OVA in the group that receive OVA in the foot pad, since the lymph node is connected. And we don't have CFSE or OVA in any of the other locations. We look in the spleen. We have CFSE in the spleen. That makes sense because both groups receive CFSE. We have OVA in the group that we given OVA intratrachely, but we don't have OVA in the group that receive OVA in the foot pad. This means that these CFSE positive cells that we are seeing here are coming from the lungs, and once they, at the same time that they are migrating, they are bringing antigen that they acquired in the lungs.
Why are the sheer numbers of the OVA cells so small?
Well, then what?
So small, the sheer numbers.
Well, the frequency is small, but the numbers are quite big because the spleen is contains 100 million cells. And when we think about the function of dendritic cells, we know that one dendritic cell, just one dendritic cell, is enough to prime a whole CD8 T cell response. You don't need too many dendritic cells to prime effective CD8 T cell responses. And I'm going to show you functionality in a minute. Okay, so if we summarize the results, this indicates that dendritic cells, when they see the influenza virus, they get activated, upregulate CCR7, they are able to enter the lymphatics, and if they don't encounter CD8 T cells, they go back into the spleen. So to finally prove this model, what we decide to do is deplete dendritic cells in the lungs, just in the lungs, and see how that affected the presence of these cells in the spleen. And we're going to deplete dendritic cells only here, not anywhere else. So to do that, we took advantage of the CD11 CDTR mice. Those are mice in which the dendritic cells express the diphtheria toxin receptor. And in these mice, we know that if we administer diphtheria toxin, the dendritic cells are going to die. So we infect them with the influenza virus. 48 hours, we give them OVA, Alexa 647, so this is an antigen. And then at the same time, we treat them with diphtheria toxin and intratrachely. So what we are doing is that we're going to have white-eyed mice that are not going to be affected by diphtheria toxin. And then in these mice, the diphtheria toxin is going to kill all the dendritic cells that are in the airways, but only the dendritic cells that are in the airways. And indeed, this is the spleen. As you can see here, this is our control group. We have OVA positive cells in the control group. Those are the dendritic cells that are coming from the lungs. In the mice that receive diphtheria toxin and are CD11 CDTR, we don't have those cells. And this is the frequency, and this is the absolute numbers. However, when we look at resident dendritic cells, the numbers are fine. That means that if we deplete dendritic cells from the lungs after influenza infection, we are losing our migratory dendritic cells in the spleen, proving that what we call migratory dendritic cells in the spleen are actually migratory dendritic cells that are coming from the lungs. And so this is the model. We have dendritic cells in the lungs. They enter the lymphatics, go into the efferent lymphatic vessels, through the afferent lymphatic vessels into the lymph node, get out and go back into the spleen. So of course the question is, if they relocate into the spleen and they have the antigen and they were in contact with the virus, so they're going to be activated dendritic cells, they have the antigen that the CD8 T cells are going to recognize. That means that CD8 T cells are going to be likely to be primed by these dendritic cells. So that's exactly what we wanted to do next, is test whether or not these migratory dendritic cells. Go ahead.
Little virus particles produced by cells or exosomes are known to carry competent MHC complexes. So your model is kind of premised on the idea that cells alone are responsible for interactions, but I believe that dendritic cells can pick up hexagonals as well. So how does, have you thought about how that might fit into the model that we presented here?
So dendritic cells can take antigen straight from the virus, but usually what they do is they take the antigen from apoptotic cells. Right. So the antigen can be acquired in different ways. But with these results, another result shows is that in order to have the antigen in the lymph node, or in this case in the spleen, the antigen needs to be carried out by dendritic cells and need to be presented by dendritic cells. I don't know if that answered your question.
Well, one could imagine, for example, dendritic cells in the lung producing, I don't know, exosomes that have viral antigens complex within that MHC, which then travel via the afferent vessel to the lymph node. Is that a possibility? How does that fit your model?
I don't think that's a possibility, because what we saw is that if we block the egress of cells from the lymph node, we block the prime. I'm going to show you. We were priming and we will block the egress of the lymph of the cells. So if in your model you produce exosomes and the exosomes get into circulation and somehow they prime CD8 T cells, this will be a passive mechanism, right? They will get into the lymphatics and from there to the blood. They will not require active mechanisms. And we saw that if we block the egress by blocking by inducing the internalization of S1P receptor, we don't have priming. I'm going to show you that in a minute.
Okay.
Plus, I'm not aware of studies that shows that effective or productive CD8 T cell response to influenza can be primed by exosomes alone. I think you can expand the duration of the CD8 T cell response by exosomes, but I don't think you can initially prime the T cell response, because they need the costimulatory ligands, they need the cytokines, they need large interactions with dendritic cells. Okay, so what we did is we decided to block the egress of the dendritic cells from the lymph nodes, and then we ask whether or not that prevents priming in the spleen. So what we did is we infect mice with influenza. Then we adoptively transfer OT1 cells. Those are CD8 T cells that are transgenic. They have a TCR that is specific for ovalbumin. Then 24 hours after transfer T cells, we have 3 groups. One group received PBS. So in this group, the CD8 T cell, there is no antigen for these transgenic T cells. So T cells should not be primed because there is no specific antigen for these cells. The other group received OVA. This is the antigen that these CD8 T cells are going to recognize. So we should have priming in the spleen if our model is right. And in this group, we give them OVA and FTY. So if our model is right, the dendritic cells that are carrying the antigen will be stuck in the lymph node, and they will not be able to migrate into the spleen, and we should not see priming in the spleen in this group. So here you have the spleen and the lymph node. This is just to show you that we don't have OVA positive cells in the control group. This is the PBS group. We have OVA positive cells in the group that receive OVA, but in the group that receive FTY, because we are preventing the egress of the dendritic cells, we don't have OVA positive cells. In the lymph node, however, we have OVA in all the groups that were administered with OVA, because FTY do not prevent the migration into the lymph node. So we look at the T cells, and I have to point, this is 24 hours after administering OVA. So this is a very short time. So if we see any signs of activation, this activation is local. So there is no time for T cells to be primed in the lymph node and then migrate into the spleen. So if we see activation in the spleen, this activation occurs locally in the spleen. So this is our donor populations. These are the CD8 T cells that we transferred. As you can see here, the frequencies are similar in all the three groups. This is the mediastinal lymph node. Cells did not proliferate at this time. This is just 24 hours, so this is very early. And here you have the activase. So in the control group, we have no activated T cells. And no matter what you look at CD69 or that regulation of CD62 or upregulation of CD25, those are all activation markers. So cells, when they get activated, they upregulate CD69, they upregulate CD25, and they downregulate CD62. Basically, if you don't have cells in this region right here, it means that you don't have activated cells. In the group that receive OVA, you have activated cells, as well as in the group that receive OVA plus FTY. This is the lymph node. So this is our control. Then we look at the spleen, and as you can see here, in the group with PBS, no activation. In the group that receive OVA, we have activation. In the group that receive OVA, but the dendritic cells are unable to migrate from the lymph node into the spleen, we have much less activation. So in that, we need dendritic cells to get into the spleen to prime CD8 T cell responses. And indeed, this is a correlation between the frequency of OVA-positive cells and the frequency of activated T cells. This suggests that CD8 T cell responses to intranasally administered antigens are priming the spleen following influenza virus infection. And if we block the capacity of dendritic cells to egress from the lymph node, that prevents the priming of CD8 T cell responses in the spleen. So to confirm these results, what we did is we took another approach. What we did is we repeat a similar experiment, but in this case, we went ahead and killed all the dendritic cells in the lungs. So what we did is we have wild type or CD11 CDTR mice. Remember, in these mice, if we administer diphtheria toxin, that kills all the dendritic cells. So we took these mice, we infected with influenza, we transferred CD8 T cells at 24 hours. Then at 48 hours, we give them diphtheria toxin alone, OVA and diphtheria toxin, or OVA and diphtheria toxin, and we have the wild type mice or the CD11 CDTR mice. And here you have what's happened after OVA administration. In the control group, this just received diphtheria toxin, didn't receive OVA. You don't have any OVA-positive migratory dendritic cell in the spleen. In the mice that were wild type and received OVA, you have migratory dendritic cells in the spleen, meaning dendritic cells that are OVA-positive. But if you administer diphtheria toxin intranasally, we don't have any OVA-positive dendritic cell in the spleen. And if you look at priming, in the control group, they don't have OVA, so there is no priming. In the wild type mice that receive OVA and diphtheria toxin, you have priming. In the mice that were CD11 CDTR, we don't have priming because we were eliminating all the dendritic cells from the lungs, and that prevents priming in the spleen. So to even confirm this further, what we did is we infect mice with influenza, we give them OVA, and then 48 hours later, we sort dendritic cells from the spleen. So we took them out from the spleen and put it in vitro with T cells. And we sort either migratory dendritic cells or resident dendritic cells. Then we culture them with CD8 T cells for 3 days. And then 3 days later, we look at proliferation and activation. And as you can see here, if we take migratory dendritic cells from the lymph node, we have proliferation. That has been shown. But if we take migratory dendritic cells from the spleen, we also have proliferation. So the cells divide, meaning that these dendritic cells are presenting antigen. And if we take dendritic cells from that were resident, should we run out? We don't. And this is just a control that showed you that if we provide antigen, the resident dendritic cells can actually prime CD8 cells. So this actually shows that after influenza infection, some dendritic cells migrate from the airways, enter the lymphatic vessels, get into the draining lymph node. If they don't encounter CD8 cells, they keep going, they enter the efferent lymphatic vessels, goes into the spleen, and once they are in the spleens, because they are activated, they have antigen, if they encounter naive CD8 cells, they can prime them. And indeed, if we block the capacity of the dendritic cells to enter the lymphatics, or we block the capacity of the dendritic cells to egress the lymph node, that prevents priming in the spleen. And of course, if we deplete dendritic cells in the airways, priming in the spleen doesn't happen. Okay, so go ahead.
Is it possible for the same dendritic cell to be presenting antigen in the lymph node and then going to the spleen and doing the same thing again, or do you know that like they're coming through the tissue, kind of passing through
That's a possibility that the same dendritic cells first stop in the lymph node and then keeps going. But because the timing of this, we infect mice, we give them the dye, and then we look in the lymph node and the spleen, and we already have cells in the spleen pretty quickly. And we know that when the dendritic cells encounter a T cell in the lymph node, they establish close contacts for like 3 days or so. So that suggests that it's not the same dendritic cells. The cells that are in the spleen didn't stay long enough in the lymph node to encounter CD8 T cells. I don't know who was first.
Naive question, but you said earlier, I think you said just a single dendritic cell can prime
Yep.
a big, I don't remember the number you used, but let's
Yeah.
say 1 million T cells. So if they stay in contact for that long, how do they do that?
No, so it's not like the same dendritic cells go and prime multiple T cells. Dendritic cells prime 2 or 3 clones, and those clones span. And at the end, you have overall expansion of the CD8 T cell response.
So, do you think it's a numbers game that they're it's random that some dendritic cells will proceed from the lymph node onto the spleen, or do you think there's something specialized about the dendritic cells that kind of make them almost maybe bypass the lymph node, not spend as much time there, and go on to the spleen?
Yeah, that's an important question. We don't know. We don't know if it's just random, that if they go into the lymph node and they don't encounter T cells, they keep going, or we have two different subsets of migratory dendritic cells. Because let me go back to this. I was trying to oversimplify a little bit, but when you look in the lymph nodes, here, this is the capsular sinus, and this goes around the lymph node. So when you have dendritic cells, the dendritic cells get into the lymph node through the subcapsular sinus, and there is two options. They migrate from the subcapsular sinus into the lymph node, and they can go around, and if they go around, this is connected to the afferent lymphatic vessels, and they can get out. So there is two possibilities. If the dendritic cells that get from the tissues in here get into the lymph node, they're going to encounter T cells, or if they don't express the right chemical receptors or they express another chemical receptors, they keep going around and they leave. And we don't know which ones.
Maybe they have slightly less expression of the sphingosine receptor or something like that.
Yeah, or less expression of CCR7 or any other chemical receptor that we don't know about. And we know that for T cells, that's the case. If T cells get in here, if they see antigen, they get in. If they don't see antigen, they keep going around and they leave. Okay, so now I need to go back to...
Like a quick technical question.
Go ahead.
So with these flow experiments, is each experiment one? You said there's like 100 million cells in the spleen. So when you want to sort spleen cells, is it one spleen or do you like group several mice?
Usually you have to group several mice just because the efficiency of the sorter is not 100%. So you lose a lot of cells. So each experiment for the in vitro priming experiments, you pull like 3 spleens and then you do that 3 times. So at the end you have triplicators from different pools.
Possible in some mice, since the number is so low of these migrating dendritic cells, it's possible that some in that group, for example, maybe don't have them, but still mount a response, or that's not possible?
Well, before pulling the mice, we always look at whether we have migratory, we do all controls, and every time that we do an experiment, we know that all the mice are infected and all the mice, if we are given CFSE, all the mice are CFSE positive. Plus, when you sort the CFSE. We sort migratory dendritic cells, truly migratory dendritic cells, CFSE positive, class 2 high, blah, blah, blah. Okay, so the question is, if we have priming in the spleen, is that important? What that means? It's just that we have priming in the lymph node and then a little bit in the spleen and that's it? We have more of the same thing? Or there are qualitative differences between the spleen and the lymph node prime cells? So when you think about a CD8 T cell response after a vaccination against tumors, pathogens, or whatever, the ideal situation is that you have a local response in the target tissue. This will be, for example, where your pathogen is or where your tumor is, so that the cells can go there and eliminate the tumor or eliminate the pathogen. And usually this is done by 2 flavors of CD8 T cells that we call effector CD8 T cells. Those are programmed to get from the lymph nodes right into the tissue. or also tissue resident memory cells, which are CD8 T cells that after encounter the pathogen in the tissue, they stay there, waiting for the pathogen to come or the tumor to come. And this is what we call a local response. So it goes from the draining lymph node into the target tissue. But in addition, we want to have a systemic response. In addition to have an immune response exactly where the pathogen or tumor is, we also have to have some prime cells that distribute along the other draining lymph nodes. So in case we have, for example, metastasis or the virus enters through another location, we have CD8 T cells waiting for them in the corresponding draining lymph node ready to fight. That's what provides protective immunity. And this is usually protective immunity systemic response is provided by central memory cells. Those are cells that are programmed not to go into the tissue, but to go into the other lymph node or the other secondary lymphoid organs. We still don't know how is that once CD8 T cells encounter dendritic cells, how it's possible that you end up with so many different flavors of CD8 T cells. And the mechanisms are still being explored, and there is no clear mechanism that explains how is that we end up with different flavors. But recent studies suggest that this early differentiation starts very early after infection, within the first 24 hours, meaning that they start after interaction with dendritic cells. And this is because people have been doing single-cell studies and looking at the transcriptional signature that correspond to the different flavors. You can already see them very early after infection, suggesting that this happens when CD8 T cells encounter dendritic cells. And of course, it could be that depending on where are you primed, the environment is different, or which subset of dendritic cells you encounter, that the dendritic cell is going to provide different signals, and you're going to become one or the other. And indeed, in agreement with it, I was talking about migratory dendritic cells, but actually there are different subsets of migratory dendritic cells. We have two main subsets of dendritic cells that are coming from the tissues. One subset is what we call CDC1, and the other subset is CDC2. And you can identify it by flow cytometry. Bottom line is that these two subsets of migratory dendritic cells are different. For example, CDC1s produce large amounts of IL-12. They express high levels of CD24. And we know that CD8 T cells or CD4 T cells that encounter these cells usually differentiate into effector cells. And the CD8 T cells or the CD4 T cells that encounter these cells acquire more a central memory phenotype. So it's possible that when you have T cells in the lymph node, if they encounter this subset, they become effector cells and they go into the tissues. If they encounter these cells, they become central memory cells and they go all around. What is interesting is that when we look in the lymph node, and this is gating on our migratory dendritic cells, the CFSC positive. So this is gating on the cells that we know are coming from the lungs. In the lymph node, you have both populations. When you look in the blood or you look in the spleen, you only have CDC2s, the CD11B positive cells. And so if you look here in this model, what this suggests is that from the lungs, you have two populations getting into the lymph nodes, but only the CD11B positive cells are able to relocate from the lymph node into the spleen. So if you think about this, what this suggests is that if you have CD8 T cells that have been primed in the spleen, they're going to be interacted with different subsets of dendritic cells compared to the CD8 T cells that are primed in the lymph node. And we know that in the lymph node, because it's very close to the tissue, the inflammatory environment is going to be completely different than from the inflammatory environment that you have in the spleen. So cells are going to be primed by different cells in different locations, and the environment is going to be different. So it's likely that the progeny of the CD8 T cells that are primed in the spleen is going to be different from the progeny of the CD8 T cells that are primed in the lymph node. So we decided to look at this very quickly. So what we did is we transferred OT1 cells. Then 48 hours, we give them OVA or PBS. So we have cells that are going to be primed by OVA. And then we look at transcription factor that we know that controls T effector differentiation versus central memory differentiation. For example, TBET cells that express high levels of TBET normally differentiate into T effector cells, and cells that are BCL6 positive normally differentiate into central memory cells. And here you have, this is gate on activated CD8 T cells. If we gate on control cells, those are, in control mice, those are mice that didn't receive OVA, so those cells are naive cells. The CD8 T cells that are not activated, They don't express TBET, they don't express BCL6, and they don't express other transcription factors like BLIM. If you gate on the cells that were primed in the lymph node, as you can see here, they express high levels of TBET, and they also express high levels of BLIM, suggesting that they are in the way to differentiating into effector cells. Whereas the cells that are primed in the spleen, they express low levels of TBET, but express high levels of BCL6. So this is an indication that you have priming in the spleen and in the lymph node, but the cells behave differently, whether they are primed in one or the other location. So, and this is the last experiment I'm going to show you. This is kind of a complicated experiment, but it's very cool. So let's see if I can explain it well. We infect mice with influenza, then we transfer OT1 cells, and then we give them the antigen. And then 24 hours after giving them the antigen, so very early after infection, we sort OT1 cells from the lymph node or from the spleen. So we sort the activated cells. So we have cells that were activated in the lymph node and cells that were activated in the spleen. So we took those sorted cells and we transferred them back into mice that are infected with influenza and receive OVA. So we have antigen-experienced T cells that were transferred into another recipient mice. They have the antigen, and then we look at day 6 after infection. So what we're going to be looking here is what's happened to the cells that were primed in the lymph node compared to the cells that were primed in the spleen. Are they identical or are they different? So this is just to show you that when we look at different locations, lung, lymph node, and spleen, we have donor cells in all the groups, and the frequencies are the same. So no matter whether you were primed in the lymph node or the spleen, you have the same capacity to get into the different locations. They don't have homing issues or differences. But this is the more important part. So I told you about the effector cells, central memory cells, and we can distinguish them by flow cytometry. And I'm not going to go into how we use it, but we can use flow cytometry to distinguish all the different subsets. So if you take, these are the mice that received the CD80 cells that were primed in the lymph node. And here you have the mice that received the CD80 cells that were primed in the spleen. And we are looking in the lungs. And if you look at this flow plot, you can see that they are pretty much identical. And they have a phenotype that corresponds to effector cells. And this is the frequency within the donor cells. And as you can see here, they are identical. The majority of cells are effector cells, no matter what. They are coming from cells that were primed in the lymph node or cells that were primed in the spleen. So if the cells get into the lungs, it doesn't matter where they were primed, they become effector cells. But something different happens in the lymph node and the spleen. And for example, I'm going to be focused on the spleen, although the same answer you get in the lymph node. What's happened is that if you take cells that were primed in the lymph node, and then you look at the spleen, the majority of these cells are what we call effector or effector memory precursors. So the cells that were primed in the spleen, in the lymph node, they differentiate preferentially into effector cells. Whereas the cells that were primed in the spleen, they differentiate into effectors, but also into central memory cells. So this is a little bit complicated, but what this means is that the cells that are primed in the lymph node are poised to become either effector or effector memory cells. No matter where they go, they are prone to be that. Whereas when you are priming the spleen, you have a higher degree of plasticity. So if you get into the effector tissue where you have a lot of inflammation, you become an effector cells. But if you go into a secondary lymphoid organs where the inflammation is lower, you have the ability to differentiate into central memory cell. So this brings us a model in which we have the infection in the tissue. Migratory dendritic cells migrate into the lymph node. We have two subsets of migratory dendritic cells. In there, you have some priming of the CD80 cell response. CD80 cells differentiate into effector or effector memory precursors. They get back into circulation, and from there they get into the tissue. Some dendritic cells, mainly CDC2 dendritic cells, can keep going and they go into the spleen. And the cells that are priming the spleen, they have a higher degree of plasticity. So if they get back into circulation and they get into the tissue, they differentiate into effector cells. But if they go back into non-draining lymph nodes or other secondary lymphoid organs, they have the ability there to differentiate into central memory cells. And they can go and colonize all the other lymph nodes, suggesting that the central memory response that provides systemic protection is most likely to be primed in the spleen. So with this, I want to acknowledge Megan, who actually worked really hard while I'm having coffee comfortable in my office. So there's one student who actually did this work, and of course other people in my lab who works really hard every day. My collaborators, Beatrice, Amy Wyman, Troy, Fran, Alex, of course, Ken, John C. at the NIH, and of course my funding agency. I will take questions. Thank you.
Thanks very much, Andre. Later time, we just have a couple quick questions and then people can come up, but go ahead. You really look like you want to ask a question. Go ahead.
It's a quick one. So the CDC1 and CDC2 cells, which are the populations of migratory dendritic cells, are they interacting with the antigen in the same way when they're in the tissue? Do they have different affinities between those two populations?
They are so yeah, there are many differences about this. So the CD103 positive cells, or are the CDC ones, they are close to the epithelial cells, so pretty close to the airways. The other ones are in the lung parenchyma. So studies have shown that the CDC ones, because they are close to the airways, they interact directly with the antigen, with the virus, and sometimes they can get infected. Whereas the other ones that are more deep into the lung parenchyma, they have access to the antigens by capturing dead cells. So they differently encounter the antigen, and they activate differently, they produce different cytokines, and they have different effector functions. And actually, CD103 positives, they die after 3 or 4 days, they start dying, and probably because they are infected.
Yeah, so I think the model you present works well for influenza infection, but I'm trying to think of other maybe more systemic infections or even other types of viral infections.
Yeah.
You know, having a different environment in the spleen versus lymph node wouldn't work for.
No, for
all these
LCMB,
infections.
for example, for LCMB, where you have the virus straight in the spleen, the resident dendritic cells are going to present the antigen there. Then you're going to have your local inflammation in the spleen, so it's going to be completely different. This only applies for infections where the virus do not get systemically, which I have to say is the majority of the infections. So LCMB, although it's very widely used in immunology, it doesn't represent a good, a normal infection.
But there still must be another way of developing memory cells outside
Yes,
of.
yes. And we are not saying that you cannot get central memory cells from the lymph node. You have to probably wait longer to get central memory cells if you are priming the lymph node.
Oh, that's interesting.
But in the spleen, you can, this is after 6 days, you can already generate central memory cells. So probably when you look at the non-draining lymph nodes, we think that most of the cells that are there, and we know that you can get them there after 3 days, 4 days of infection, are coming from the spleen.
Okay, great. I think a later time we'll call it now, and there are a couple other questions. Please join me again in thanking Dr. Robinson.
Thank you.